Hi, welcome to my YouTube channel Fast Track Learning. In this video, we will talk about cell cycle checkpoints. You can see in the figure, there are three checkpoints. First of all, we have G1 checkpoint, then we have G2 checkpoint, and third one is M checkpoint. The G1 checkpoint is between G1 and S phase. Then we have G2 checkpoint, which is between G2 and M. And then we have M checkpoint. First of all, I'm going to tell you what is cell cycle. Cell cycle is the series of events that takes place in a cell that leads to its division to produce two daughter cells. Actually, cell cycle is the process by which a cell grows, it copies its DNA and divides into two new cells. Why cell cycle is so important? The cell cycle helps organism grow. It helps to heal wounds, replace old or damaged cells. So cell cycle is very important. Okay, there are three states of cell cycle. They are mitosis, quiescent, and interphase. First of all, we have interphase. It include G1 gap phase, S phase which is known as the synthesis phase, then we have G2 phase which is known as gap 2 phase. Interphase is also known as preparation phase. The cell spend most of its time here. It has three parts. What is G1? What is gap 1? In gap 1, the cell grows bigger and make proteins. In S phase, in synthesis phase, the cell copies its DNA, so each new cell will have the same DNA. In G2, the cell grows some more and prepares to divide. Okay, now we have mitosis. Mitosis states this is the division phase. This is where the cell actually divides into two new cells in mitosis, and it has four steps which are prophase, metaphase, anaphase, and telophase. Now, what is prophase? The DNA forms tight bundles called chromosomes in the prophase. What happened in metaphase? The chromosomes line up in the middle of the cell in metaphase. What happened in anaphase? The chromosomes are pulled apart to opposite sides of the cell. Telophase. Two new nuclei form, one from each new cell. Also, there would be cytokinesis, which is known as the final separation. In this, the cell cytoplasm split and formed two new cells, each with its own nucleus and identical DNA. There is also G naught phase, which is resting state or gap phase or quiescent state. What happened in G naught? After exiting mitosis, some cells remain in G naught phase. What are these cells? These cells are nerve cells, red blood cells, heart cells. They remain in G naught phase. Now let's talk about the checkpoints in cell cycle. First of all, we are going to compare the checkpoints with the traffic signals. We can compare the checkpoints in the cell cycle with traffic lights at a busy intersection. The traffic lights are here controlling the flow of cars, you can see, to ensure safety. So we can compare them with the checkpoints that are controlling the flow. They are controlling the cells. They ensure that there would be proper cell division. So let's study more about checkpoints. What are actually checkpoints? Checkpoints are actually the regulatory pathways that assess whether the cell is ready to proceed to the next phase. If the cell is ready to proceed to the next phase, then the cell cycle checkpoint will allow the cell to enter into the next phase. Otherwise, it will not allow. Checkpoint is a stage in the eukaryotic cell cycle at which the cell examine internal and external cues and decide whether or not to move forward with division. If internal and external cues allow, then it will allow to move forward with division. But if the internal and external cues will not allow, if the conditions are not good, so it will not move forward. Basically, the purpose is to explain what is cell cycle, what are checkpoints, why they are crucial for maintaining cellular health, and how it prevents different diseases like cancer. Also, checkpoints have function in DNA repair, cell growth, and ensure that damaged or incomplete cells do not divide. These are all the function of checkpoints. So what is the first checkpoint? The first checkpoint is known as G1 checkpoint, which is present between G1 and S phase. The G1 checkpoint is at the G1 to S transition. The main decision point for a cell, which is G1 checkpoint. This is the primary point at which it must choose whether or not to divide. Once the cell passes the G1 checkpoint and enter into the S phase, it becomes irreversibly committed to division. So if it once it crosses the G1, once it enter the S phase, it have to divide. That will cause unexpected problems like DNA damage or application errors. A cell that passes the G1 checkpoint, it will continue the rest of the way through the cell cycle and produce two daughter cells finally. But if there is genome damage, so entrance into S phase is blocked due to the G1 checkpoint because it is located at the end of G1 phase before the S phase. Checkpoint will check, it has a function that it will check the cell size, it will check the DNA integrity, it will check the nutrient availability before proceeding to the next phase. We can take an example. It's a relatable example like a student checking if they have all the materials, there is no any kind of problem, absolutely ready before an exam, enough resources to proceed. So it's like that. If there is no genome damage, nutrients are available, DNA integrity is there. So only in that case it will allow the transition from G1 to S phase. Otherwise, there is no permission. So this is the main function of G1 checkpoint. So that is why it's called the main decision point. This is the primary point. Also, there would be the consequences of failure. Failure, if takes place at the G1 checkpoint, then it can lead to mutations. It can lead to cancer. Let's talk about the second checkpoint, which is G2 checkpoint. What is the location of G2 checkpoint? It is located at the end of G2 phase before M phase. The G2 checkpoint is at the G2 to M transition. Function, it checks if no DNA damage, if there is DNA damage or not, and ensures all DNA is replicated correctly. We can take an example, we can compare it to proofreading an essay before submitting. We ensure that there must be no errors in our writing material before submitting. So it like that the G2 checkpoint check if DNA is replicated correctly or not. The cell will check DNA integrity. Is there any of the DNA damage or not? Also, it will check DNA replication. Was the DNA completely copied during S phase? If errors or damage are detected, the cell will pause at the G2 checkpoint to allow for repairs. It will not proceed. Instead, it will allow for repairs if there is a damage. If the checkpoint mechanisms detect problem with the DNA, the cell cycle will be halted and the cell attempts to either complete DNA replication or repair the damage of the DNA. If if the damage is irreparable, the cell may undergo apoptosis or programmed cell death. This self-destruction mechanism ensures that damaged DNA is not passed on to daughter cells and is important in preventing cancers. So, it is doing two things at the same time. If there are the chances of repair, it will help to pause the cell cycle so that damaged DNA would be repaired. But if there are no chances of repair, if the damage is irreparable, the cell may undergo apoptosis or programmed cell death. This is known as self destruction. It ensures that damaged DNA is not passed on to the daughter cells. Otherwise, what will happen? What will be the consequences of failure? If the checkpoint is not be able to work correctly, if the G2 checkpoint will not function correctly, so what will be the failure consequences? In case of skipping this checkpoint, it can lead to cell division with damaged DNA, which contribute to tumor formation. So, if there is a failure in G2 checkpoint, then there will be tumor formation. Now let's talk about the third checkpoint, which is M checkpoint, which is also known as spindle checkpoint. This is the last third checkpoint. M checkpoint is known as spindle checkpoint. Here the cell examine whether all the sister chromatids are correctly attached to the spindle microtubule, because the separation of the sister chromatids during anaphase is an irreversible step. Once there is separation of sister chromatid takes place, this is irreversible. The cell cycle will not proceed until all the chromosomes are firmly attached to at least two spindle fibers from opposite poles of the cell. So, this is very important checkpoint related to this. Okay, in this figure we can see here that there are attachments, there are sister chromatids that are correctly attached to spindle microtubule. So specific age is important in metaphase. Now we will talk about what we discussed, that what is the function of M checkpoint, what is the function of spindle checkpoint. It ensures that chromosomes are properly aligned and attached to the spindle fiber before separation. So we can see in this figure that chromosomes are properly aligned and they are attached to the spindle fibers before there will be separation takes place, that will lately takes place in anaphase. To understand this, we can use a sports analogy like making sure that all the players are in position before the game starts. Here we can see all the players are there in their specific position before the game start. So like that, all the sister chromatids are also correctly attached to the spindle microtubule. So we can understand easily from this. But what would be the consequences if there would be failure takes place? Like errors takes place at this checkpoint. If errors takes place at this checkpoint, then it can result in aneuploidy. It can lead to conditions like different diseases will occur, like Down syndrome, cancer, mutations will take place. I hope you understand the checkpoints very well. These are the checkpoints that are in cell cycle. These that were G1 checkpoint, G2 checkpoint, and M checkpoint. We have discussed that G1 checkpoint is between G1 to S transition. It is very important because if there is a problem, if there is damage in G1, so entrance into S phase would be blocked because of G1 checkpoint. Because G1 checkpoint is checking the cell size, it is checking the DNA integrity, it is checking the nutrient availability, and then it will decide whether to proceed or not to the next phase. So it is very important checkpoint. We also discussed that what was G2 checkpoint. G2 checkpoint was also very crucial. G2 checkpoint is located at the end of G2 before M phase. Its function was to check for DNA damage, and also it checked all the DNA is replicated correctly. If all the DNA is replicated correctly, then it will allow to proceed for the next phase. But if there is a failure in G2 checkpoint, then there would be the consequences of that failure that include tumor formation. Thirdly, we talked about M checkpoint. It is also known as spindle checkpoint. In this checkpoint, cell examine whether all the sister chromatids are correctly attached to the spindle microtubules. But if there are error at this checkpoint, what will happen? It will lead to different diseases, like it can result in aneuploidy. It lead to conditions like Down syndrome or cancer. We also discussed about a sports analogy in this M checkpoint, like we make sure that all the players are in position before the game start. It also ensures that all the sister chromatids are correctly attached to the spindle microtubule, because the separation of the sister chromatids during anaphase is an irreversible step. The cell cycle will not proceed until all the chromosomes are firmly attached to at least two spindle fibers from opposite poles of the cells. So it is very important. I encourage you to learn more about cell biology and its impact on health. Also, like, subscribe, and comment with questions on topics for future videos. You can suggest me different topics for my future videos as well. Thank you. I hope you understand very well.